Well, hello everybody. My name is Philip Chapman. I'm going to be presenting the webinar today. I'd like to thank you all for joining us. I hope that you can see the visual on the screen okay and you're able to hear me okay on the audio. I would just like to request if anyone is dialing in on their telephone or is listening to the audio on their computer, please could you ensure that you mute either the microphone or mute your phone so we don't get any noise interference during the presentation. Thank you very much. Okay, if you have any questions during the presentation, we have the Q&A window that you should hopefully be able to see on your screen. You can type your questions into there and then we'll address the questions at the end of the presentation. Okay, and without further ado, we're going to begin. So what I'm going to be talking about today are really the new capabilities that we've been launching now with the NGC instrumentation and the ChromeLab software. And these address the points for speeding up the work process, for more streamlined ways to optimize methods and determine optimal separation conditions, and then also how you can bring together multi-step purification methods, and instead of doing them in the more traditional sequential way, then by bringing them together into an automated multi-dimensional chromatography method. And we'll be looking at the considerations of designing these sorts of experiments and also be looking at some examples. So the benefits of doing an automated 2D or multi-D purification are obviously the inherent speed and the convenience. If you can imagine a traditional purification that may be a 3 or 4 step purification, this could take 2, possibly 3 days to complete. When you run the first separation, you'd collect the fractions, you would analyze the fractions in a gel, determine where your target protein is, and you would then pull these fractions, dialyze them, and then load them onto the next column. And this process can be very time-consuming. There can be the potential for human error being introduced if you miss one of the fractions or you pull an incorrect fraction after you run the gel. And it also takes up a lot of your time. So what we're looking at now is a way to automate this process where you can write one method that would take you through all of the steps of the purification, and then you could just have the confidence to be able to walk away from this and leave it and come back after a few hours, and then you have your purified target protein at the end of this. Another advantage of this obviously is it maximizes the efficiency of the purification. Every time you run a sample over a column into a fraction collector, and you're running these on a gel and dialyzing, you're always losing some of your target protein at each of these steps. And so if you can remove these steps, then you can minimize the amount of sample loss that occur. To be successful with an automated multidimensional method, the heart of it is really well optimized single-step methods. When you're approaching a new purification procedure, you can't just straight away write a 5-column method and be able to run it straight off. You'd need to have done some optimization to know the binding and elution characteristics of your sample over each of the individual columns. And then from that, you can take that information and easily build it into an automated method. So the sorts of factors that you would need to know from your single-step purifications are the ones that we see on the screen here. You'd need to know the optimum column flow rates for each column for the maximizing the retention time and the binding of the protein to the column. You'd need to know when your target protein of interest elutes from each of the individual columns, in what volume it elutes from, because if you're going to elute from one column and capture that eluate into a static loop prior to having it inject onto the next column, you need to ensure that you have a sufficient volume to capture all of the eluates. Also, conditions such as the pH, if you're using ion exchange columns, and you're going to elute from one column directly onto another, you need to know that the pH conditions would be compatible with both chemistries. So in the method optimization approach, for those of you that are familiar with the Chrome Labs software, we have the scouting feature. Now this is really single step optimization. It allows you to optimize a number of different conditions to determine the optimal purification process for each column, and it allows you to do them systematically one at a time. And so we can see on the left-hand part of the slide here the conditions that can be optimized using the scouting function within the Chrome Labs software. You can optimize flow rates, where you could be injecting your sample over the column a number of different times at different flow rates, again to optimize maximum flow rate for the retention of the protein and binding to the column. The %B, so the gradient with which you elute from the column. Do you do a very sharp isocratic gradient? Do you do a gradual one? Would you do step elutions? Optimal pH if you're doing ion exchange chemistry for optimal binding and elution of your target protein. You can even scout across different columns. If you're unsure of which is the best column for the particular protein, you could do multiple scouting runs over different column chemistries. And then also the binding capacity, how much sample can I load to the column to maximize the capture without overloading the column and then having the target coming off the column in the flow through. So this slide sort of graphically summarizes and represents what I've just explained to you. From the single sample, you could then scout over different column chemistries. From that, you can select the optimal column, and you could then scout on that over different pH conditions. And then from there, you could also then scout across different elution gradients, the angle of the gradient, the length of the gradient, an isocratic or a step. These are all ones that can be scouted in an automated fashion to then quickly give you the optimal method that could be then moved forward into your multi-D methodologies. So taking these principles here, we've got an example. This was some work that was done internally, and this was to purify one of the proteins that's used in our molecular weight markers, the rainbow markers that we use for our gel electrophoresis. And so with this, we had to try and isolate the chromogenic protein that's Prancer purple, that has the purple fluorescence tag upon it. And then we were looking for the best column, the best pH, and then the best elution gradient to isolate the target. So the first step was taking the sample and running it over different anion exchange columns. Again, this was done in an automated manner, so it was using a column selection valve. The sample could be loaded, ran across the column, and at the end of the run, the method would then automatically switch to the next step and would then run the same method over a different column. And so from this, we then had the three different chromatograms that you can see on the right. These are three different anion exchange columns, and then taking the target fractions from the elution and running these into a gel. And so we're looking for the strong bands at 25 kilodaltons here. And because this is a chromogenic protein, we were monitoring the wavelengths of 280 for the protein absorbance, but also we could monitor at 525 nanometers because this would be for the purple dye upon the protein. So this could help us also in the scouting determine which is our protein of interest and differentiate that from other proteins that may be absorbing at 280. So this sort of run would take maybe 2 hours to complete over the different columns. And then using our stain-free gels, we could load the gel and then quickly run the gel and image it and determine the optimal column to then move forwards with. Having then completed the column selection, we then moved on to selecting the optimal binding and elution conditions for the protein. And so again here we've got the one column, and then in the chromatogram we can see we're looking at it over 4 different pHs ranging from pH 7 to pH 8.5, and these are in 0.5 increments. This was achieved using the buffer blending valve, which, if you're not familiar with, this is a valve that enables you to automatically prepare buffers in the system just from stock solutions at the required pH and salt concentrations. And so the buffers are prepared automatically, the run is completed at a given pH, and then the system would switch, load more sample, and prepare the buffers at the next pH required. And again, collecting the fractions, running them in the stain-free gel, we see a nice strong band of the target protein at 25 kilodaltons, and we're looking then to see where this band is strongest relative to the additional contaminant proteins. And then from this step, we selected the pH 7.5 was the best optimal pH for yield and concentration of our target protein relative to the other contaminants. And then the next step to be scouted is then the % B elution gradient. And as you know, you want to try and elute as quickly as possible to give a nice sharp resolved peak, but without co-eluting for the other contaminant proteins that are present. And so again, different runs here at different % B, final % Bs ranging from 0 to 20% B up to 0 to 50% B, as we can see here. And then we see the corresponding chromatic up here. And again, down here with the gels, we can see a nice strong enrichment of the target protein. And then we see the different levels of contaminant proteins that are also co-eluting. So to summarize this scouting approach, it's a way to quickly determine the optimal purification conditions for your target protein. And having a well-optimized purification strategy is essential to then be able to automate this into a 2D or a multi-dimensional purification procedure. So in the final gel that we're seeing here, we can follow the purification process from start to finish. In the left-hand lane, we have the molecular weight standards. The one next to that is the crude feedstock or the loading sample, where we can see the target band at 25 kilodaltons. After running through the Nuvia Q column, which was selected as the optimal column, in the flow-through, we can see a lot of other proteins being eluted, and we can see a tiny, tiny amount of the target protein. So confirming we've got good efficient binding of the target onto the column. When we then elute from the Nuvia Q, which is this lane here, we see then a very strong enrichment of our target protein, but we also see some additional contaminant proteins at around 37, around 20, and maybe around 18 kilodaltons. I skipped a lane because the one to the left of that is 100% B, the strip wash at the end of the Nuvia Q run, and this just shows that we've stripped everything off of the column and that we had successfully eluted our target protein during the gradient because we see none of the 25 kD signal and the final strip wash. And then what we next see are we see two additional columns that we used, the mixed mode CHT column to clean up the sample, and then the final size exclusion column here. So we see then a nice 4-step purification that was easily optimized the conditions for this within a single day, just using the features within the system and the software. So getting to the main content and the main focus for the webinar now. As I've said already, in the perfect world, we could take our crude biological sample and we could isolate our target biomolecule of interest just over a single column. But as we know, none of us live in this perfect world, and just through the inherent complexity of the samples that we're working on, purifications are usually considered to be multiple step purifications, where each step involves running the sample over a different column chemistry to further purify the target of interest. Now, there are lots of terms that are used to describe the technique, which are sort of Kleenex terms, if you understand. They're generic terms that can be used interchangeably, but they do have some quite specific differences. And so with this slide, I just want to try and confirm and clarify the definitions we're using here, so we're all on the same page in the meanings that we're discussing. So we often hear people referring to tandem chromatography. We often hear people talking about two-dimensional chromatography or multi-dimensional or multi-D chromatography. And these words and terms are often used interchangeably, but they are different. The key difference is that with tandem chromatography, effectively you could imagine the two columns are daisy-chained together, they're linked together. And so all of the sample is flowed over column 1, and then all of the eluate from that column is flowed directly over to column 2. And so for that to be possible, by having the two columns in line and flowing everything from column 1 to column 2, there are some very significant considerations that must be taken into mind as to whether or not this is possible. And those are the ones that are summarized on the bullet points here. Binding capacity is obviously a major consideration. If you have your first column that can bind your target at 50 mg per mil, your second column only has a binding capacity of 2 or 3 mg per mil, and you run them in line, then you're going to overload the second column, and you're going to lose your protein through the flow through and not binding efficiently to the column. If they have different buffer composition requirements, if one requires a very high salt concentration, the other requires a low salt, then the two columns couldn't be put together in line. If they have different pH requirements for optimal binding, then they couldn't obviously be used in line as well. Other considerations as well are other factors that may be in the buffers that you're using. So for example, if you're using IMAC columns for isolating a histag protein, the common factor that's used to elute from this column is imidazole. And as I'm sure a lot of you know, this absorbs at 280 nanometers. So imidazole may appear flowing through that could be masking peaks or contaminants or your target protein of interest during the elution. So these are all considerations, along with one of the very crucial ones, which is the column pressure ratings. Now the approach that we use with the NGC instruments is to do tandem chromatography. We would include 2 column switching valves or column selection valves in the instrument, and we would have 1 column plumbed to each CSV. The advantage of this is it enables you to load your sample onto the first column, and you can keep the second column offline at this point. You can wash the unbound material from your first column without running it over the second column. And then when you begin the elution from your first column, you bring the second column online, and then the eluate from the first column is directly sent over the second column. The major advantage of doing it this way and having both columns on column switching valves is you can accurately measure the column pressure ratings, pre-column and delta column pressures during the whole run. So you can be certain that you're not causing any damage to either of the columns by overpressuring them. Because if you imagine your first column is a low pressure column and your second column has a higher pressure, that's going to be generating back pressure onto the first column when you come to the elution step and you bring the second column in line. So it's important, I would say it's essential, to be able to monitor the column pressures during the run to ensure you're not causing any damage. So this is tandem. Think of tandem as linking the two columns together. Now, because of the considerations, the quite significant considerations of binding capacities, column pressure ratings, buffer composition, tandem chromatography is generally less common because of the limitations. Where tandem is often used is for affinity purification. If, for example, you're doing antibody purification and you're eluting from a protein A column, as you know, you elute from the protein A in an acidic pH, you want to quickly neutralize this so you don't denature your antibody. So commonly people would then bring a desalting column in line after the protein A column, and this can be done in tandem, so you could simply elute from the protein A column, run your antibody eluate straight through the desalting column, which effectively is your buffer exchange step, so you can immediately neutralize your antibody as you bring it off the protein A column. So for more complex purification procedures that are more than just a simple two-step, this is what is often then called two-dimensional chromatography, or if there are more than two columns, it's multi-dimensional chromatography. And the key difference with this is that the proteins are eluted from the first column, and instead of being shuttled directly onto the second column, they're eluted into some sort of static loop or sample vessel. They're captured in there, and then that sample from the loop can then be directed onto the second column. So by taking the second column out of line during the elution step, your considerations about column pressure, buffer compositions, binding capacities, etc. are negated because you're not going directly from one column to the other. And so these 2D or multi-D techniques are what are more commonly used. So for those of you that are familiar with the NGC instrument and the software, you will have seen the method templates that we have built into here. Traditionally, historically, one of the complexities of being able to do 2D and multi-D chromatography has been the complexity of creating the methods and ensuring that what you're eluting from one column is being directed to the next column in the correct way, the correct volumes, the correct buffers are being selected, etc., etc. So what we have built in within the software are a range of different templates, and you'll see with each release, future release of the ChromeLab software, you'll be seeing more and more methods being put into these template folders. What it also has is it also has a little graphic, as we're seeing in the lower panel of the slide here, which is a graphical representation of what is occurring during the purification run. So just to explain what we're seeing here, we're seeing we have two affinity columns on one column selection valve. We're loading via the sample pump, we're sequentially loading two different samples onto these two different affinity columns. That's what's represented in the left-hand side here. After the loading is completed, we can then sequentially elute from the affinity column and then elute from there and capture the target into the static loop. And as I'm sure a lot of you know who are familiar with doing affinity chromatography, you can often load the sample in the forward flow, wash away the non-binding and non-specific proteins, but then if you elute in the reverse flow, it gives you a much more concentrated packet or band, if you like, of your target protein. So and again, that's what's being represented here, where we can elute from the affinity columns in the reverse direction into the static loop. And then the next step that we're seeing on the right-hand side is we're then seeing we can load the sample directly from the loop over, in this case, a size exclusion column, and then from there into the detector and the fraction collector. So we don't show the elution profiles, which we show in the other method templates, because it's more complex here because we have multiple samples, multiple columns. So instead you see this graphical representation of what is actually going to be occurring during the method. There's also in the notes section a little place you can click on, and this is a useful tool really that just summarizes what's going to be occurring during the method. It gives you the system requirements of what components you would be needing for this, just to be able to clarify and visualize in your mind what's needed before you start the run. So also what we have within the ChromeLab software, because you're now using multiple valves that have the same function, you could be using 2 or 3 column switching valves, you could be using 2, 3 inlet valves, some could be as buffer inlets, some could be as sample inlets. What we then have now is when you come to configure the instrument, there is the fluidics mapping scheme. And what this does is this helps you with the placement of the modules within the instrument, and it also labels them so you know which is column switching valve 1, which is column switching valve 2, etc., which is a buffer inlet valve and which could be used as a sample inlet valve. So again, this just simplifies the configuration of the instrument, and so you know during the method where you should be plugging which columns in, where you would be loading your samples through, etc. And this can be used in conjunction with one of the new pieces that we've launched now that you can find on the website, which is the plumbing guide. And again, this is just designed to help you, to enable you to successfully plumb the systems. And what this plumbing guide has is it shows multiple different instrument configurations, and it shows the pre-written method templates that can be used on these configurations, and it highlights, if you like, the non-standard, non-pre-plumbed tubing that you would need to add to your instrument to then be able to run these methods. You can also see on the Chrome Labs software document DVD or on the website, you can see the animations that talk you through and show you the tandem and the 2D methods. And again, just to show you the fluidics flow path, to show you what's going where at each stage of the method. And again, just trying to simplify, just trying to ease the comprehension and ease the adoption of these techniques. So having sort of spoken about the principles now, I'll now show you a couple of examples of this, of some very common purification methods that people do globally. The first one is a simple antibody purification. Now, I did this many times in a former life before I joined Bio-Rad, and the top traditional workflow is the one I've done many, many times. With this, you load your sample onto a protein A column, you would loop from the protein A column in an acidic buffer, you then neutralize the fractions to avoid denaturing the antibody. This would then need to be diluted into a different buffer to make it compatible with the next column, which would then be the ion exchange column. After the ion exchange column, you could then collect the fractions, and you'd run then the antibody over your size exclusion column to remove any aggregates or to remove any heavy light chain fragments. Now, this process was a 2 to 3 day process when I did this routinely, with the dialysis, with the gels, etc. included in here. So quite a lengthy, laborious procedure. So with the automated procedures that I'm about to show you, this uses 4 columns run using sequentially in an automated method, where we run from the protein A column, we elute from the protein A column, again in the acidic buffer. This runs straight over a desalting column, which buffer exchanges into the buffer required for the ion exchange column. The eluate is then directly injected over the ion exchange column, and from there it can be passed over the size exclusion column, again to check for heavy light chain fragments, to check for aggregates, etc. So what we're seeing here is a graphical representation of the fluidic scheme that, if you have the Chrome Labs software, you'd be familiar with. And this is showing how the instrument was configured for this example that we're going to show you. We had with this buffer inlet valve, so we could switch automatically in the method between the buffers that were required for the protein A column, the buffers that were required for the ion exchange column, and the buffers that were required for the size exclusion column. We can use up to 2 inlet valves, sample inlet valves. So this means that once you have this method written, not only is it automated for a single sample, but you can repeat this process time and time and time again for multiple samples. And you can load through the sample inlet valves through the sample pump, and you can also have one line as the rinse buffer. So between sample loadings, again, you can just easily program it to flush the lines, flush the pump, and have it ready then to receive the next sample. These can also be set up with air sensors, so it can become more truly unattended, where when it detects the end of the sample, it would just then automatically move over to the next step in the method. The use of an outlet valve then becomes essential, because this can direct the materials that you don't want directly to the fraction collector or to waste, and it can direct the eluate that you do want back into the injection loop where it can then be directed to the next column or into a static loop if it's an intermediary capture step. And again, with multiple column switching valves, you then have high capabilities of throughput for the number of columns that you can use and the number of sample loops that can be used in the intermediary steps. So what we're showing here is we're showing the chromatogram at the end of this run. This is actually skipping the ion exchange column, which I'd spoken about previously. That's in a separate example. But what we have here on the left-hand side, this was all done as an automated 2D or multidimensional purification method. So the methods were written in the same template for the protein A column, for the P6 desalting column, and then into the size exclusion column. And what we then see is we see here, to begin with, the unbound protein from the protein A column. This will be directed to waste. We then have the elution gradients from the protein A column as seen here by the %B profile and as seen here by the drop in the pH. But we don't see the elution peak from the protein A column. And the reason that we don't see this is that this wasn't directed through the detector. This was sent to the P6 column, which was plumbed in line. So this first step was a tandem step where we eluted from the protein A and sent it directly into the P6 column. So that's why we have the peak missing from the protein A elution. What we then do see is we then see the peak from the desalting column. So this went straight from protein A to P6. After going through the desalting column, it went to the detector, which gives us a signal. And then the eluate that we were interested in was then directed back from the outlet valve through to a static loop connected to one of the CSVs. And then from there, because we had a large volume from the desalting column, we could then do sequential injections of this onto the size exclusion column, because we couldn't go directly from the desalting to the size exclusion just because of the capacity of the size exclusion column. So again, for those of you unfamiliar with ChromeLab software, there are lots of nice tools within the software. As we see down here, when you're collecting the fractions, it's nice to have a fraction heat map. This very quickly shows you which tubes in your rack contains your proteins. And the intensity of the coloring for each tube corresponds to the amount of protein that's present within those tubes. So again, it just quickly, you go to your rack, you have 96 tubes, you look at the heat map and you see straight away, these are the tubes that I need for my next step at the end. Not only was this done just to show the proof of principle, but we also did this using a human monoclonal antibody that was expressed in a Chinese hamster ovary cell line. And we were looking at this at varying degrees of aggregation within the antibody. So the same procedure as before, protein A directly onto desalting column. From the desalting column captured into a static loop. A small amount of that was sent over the size exclusion column to analyze for aggregation, fragmentation, and then the rest was directed from the loop into the fraction collector. So again, we see the same chromatogram on the top part of the screen here. And what I'm showing highlighted on the bottom part of the screen here is just the peak from the size exclusion column. So what we see here on the right that I'm showing with my cursor are the 3 monomer peaks. These are 3 separate runs overlaid. These are 3 separate runs overlaid at the result after the sample is passed over the 3rd column. So you see, even after running the sample over multiple columns and doing this multiple times, this is a very reproducible technique. What we see to the left of the monomer peak is then the aggregate peak, and we can see here the differentiation between the different levels of aggregation. So this is reproducible, it's consistent over multiple columns, and it still gives you good analytical data to be able to differentiate and determine relative levels of aggregation of the antibody from a CHO cell line. Okay, hope everyone's still interested. We're in the home straight now. The next slide is somewhat of a more complex example. Again, this is a multidimensional purification, and it's taking a traditional IMAC purification method and taking it from the sequential single steps and building it into a multi-D template and a multi-D method. And so again, summarizing the workflow at the top, the IMAC column, dialyzing the fractions, analyzing them in a gel, moving on to the ion exchange column, collecting those fractions, analyzing them in a gel, and then taking the fractions of interest onto the size exclusion column. So taking that sequential step method that could be 2 or 3 days, allowing for the dialysis and gel run times, and then bringing it into an automated process that took 5 hours from start to finish. Again, with this, optimized methods are crucial. So before we did this, we did some simple scouting steps. We did scouting across the IMAC column to see the best conditions to elute from the IMAC column. We were here scouting over different pre-elution washes of the column with the imidazole, just to find the best way to remove the non-specific proteins, but still isolate our target protein. So this protein was expressed in an E. coli cell line. It's green fluorescent protein, GFP, sumo protein. So it has a traditional 6-his tag. It also has a green fluorescence tag upon it. So again, we were measuring this at 2 different wavelengths. We were measuring at 280, which is our protein signal, and we were measuring at 495, which is the signal from the fluorescence tag. And again, this helps us because it helps us identify which is our protein of interest. And also, as you know, for those of you that's done IMAC, when you elute with imidazole, imidazole absorbs at 280. So this can also give a signal in the elution phase that doesn't correspond to protein. It's just simply imidazole. So having the 2 wavelengths here just helped us determine which was our peak of interest. So having scouted and optimized for the IMAC column, we did a simple pH scouting over the ion exchange column. Here we're showing just 3 different pHs from that, just to determine which was the optimal pH for binding of this particular protein to our Q column. Having determined that, different elution gradients from 0 to 20 up to 0 to 70% P. Again, just automated optimization steps, just to make sure we had the best methods for each column to then bring it together in an automated process. This is a representation of the fluidic scheme of the instrument that was used, again with 2 column switching valves, one holding the columns, one holding the loops, and the outlet valve after the detector to help us direct our proteins of interest back to the loops to then go to the next column. So don't be scared by this. This was a method that was created in ChromeLab software. For those of you that are familiar with the templates for 2D, they're 2-step. They are true 2-dimensional templates at this time. But what I'm showing here was you can easily modify and customize these templates, use them as the building block to add additional phases. And that's all we did here. In the left-hand side here, this is just the prepping the columns, prepping the lines, prepping the system. So this is just flushing the loops, equilibrating each of the columns, making sure it's ready. We could then bring in phases for the IMAC column, for the application and elution. We could bring in another phase for the P6 column. From here, we could then bring in another one for the ion exchange column, and from there, another one for the size exclusion. And then again, at the end of it, we can wash our instruments and leave it nice and clean to sit overnight. So it's very easy to use these templates as the building blocks to then expand much more complex methods from there. Now, what I personally really liked by this was none of this was done in tandem. When you do tandem, you go straight from one column to the other. You don't see the peak until the end of the purification. So there's that trust issue, there's that leap of faith. Is my target going to be there at the end? So with this one, we set it up so that after each one was a multi-D where the protein would be eluted from the column, directed through the detector, The target protein of interest was then shuttled into a static loop, and then from the loop it was directed into the next column. So that means that at each step, even though this is fully automated, I can press start and walk away, I can come back and just check. It's that reassurance that yes, this is fine. So running over the IMAC column, the elution from the IMAC column here, this was then into a loop and then injected from the loop over the desalting column. The peak was then directed into another loop. From there it was injected onto the ion exchange column, was eluted from there through the detector, so again we know our protein peaks there, into another static loop, and again from there just through the binding capacity or capability of the size exclusion column, multiple injections from the final static loop over the size exclusion column. And this just shows the data again, but showing here the recovery values at each step. So you can see the peaks, and you can see, again, this is a 4-column, fully automated method with excellent recoveries at each step. And this is recovery calculated from peak area. And in this, we calculated the peak area from the 495 nanometer signal, which is the brown trace, just because the 280 was less reliable because of the presence of the imidazole. Because again, the imidazole absorbs at 280, so it gives a less accurate peak area calculation. So a lot of this is about confidence. It's about the comfort and trust in knowing that I can create this program and I can trust it to run. Even though I'm not running a gel after each fraction, I still know that it's there at the end of it. Again here, some of the tools, the data analysis tools, the single-click analysis, the information that you can get very quickly from the NGC, single chromatogram analysis, multiple chromatogram analysis, easy overlays, easy integration, etc. The heat map for the fraction rack, so at the end you know quickly where is my sample. The protein concentration calculations that can be done within the software that then give you the sort of the amounts and the purity of each. But as I said, it's trust. Anyone who's done a lot of chromatography, you stand close to your system. We've all spent hours and hours and hours in the cold room nursing our protein through the system. But we have the tools now. Things have moved on. We have the tools and the capabilities to automate a lot of this. You know, if you've spent 7 hours of your day in the cold room nursing your purification, and you could now set this up to run automated, press start, you've got 7 hours of your day back. Just think what you could be doing with all of that extra time. So some of the other features within the software, you know, we have the air sensors. Air sensors are great for setting up automated runs because they will detect the end of sample and switch automatically to the next stage for you. If they detect end of buffer, it will stop the run. You're not going to kill your column by pumping in air anymore. With the ChromeLab software, if your system's networked, you can set it up that it will even email you at the end of the run. You know, how many of you, I can't see you, but I'll ask you to raise your hands, how many of you have started a run at 5 o'clock in the evening, gone home, and you've crossed your fingers, hoping that when you come in the next morning, it's not just a puddle at the bottom of the fraction collector, or it didn't stop 5 minutes into the run because it ran out of buffer. It will email you. It will tell you that the run's completed. If there's a problem, it will notify you as well. We have within there featured as well, you're doing this lots and lots. How good's my column running? There's the column performance test within the software, where after 2, 3, 4 runs, you can run this routinely just to confirm the column's still binding well, it's still performing as well as it should. And these are easy things that are pre-programmed in the software that you just drop into a method. And at the end of it, you could have, with the buffer inlets, you could have one line going into your wash solution. At the end of the run, it will automatically put the columns in bypass and it will flush through the systems. How many of you, again, with your virtual show of hands, have gone to your system the day after someone else, and they left it in the 100% B, and there's salt precipitates everywhere. We can eliminate those risks. It's just, I get passionate about this because I've suffered this in the past. It's just having that confidence that now this is on my side. This is a tool that can help me do what I want to do. So I've talked for more than long enough. I thank you all for your attention. We'll now look at the questions that you've been writing to Candice. Okay, so we have a question about some of the literature that we've been talking about. The plumbing guides is available from the website. We have application notes, app notes that you can find on the website that also talk through in a little more detail the examples that we're showing. I would say either contact your local representative if you have any questions. You can contact Candice or myself directly, or you could just go to the website and look on the website. You can find out more information about the software, the hardware, the applications, and these tools. If you're interested and you have an NGC and you want to start to do this multidimensional chromatography, please get in touch with us and we can help you. We've got lots of tools to make this easier for you. Okay, I'm not seeing any additional questions popping up now. So in that case, I'd like to thank you all for your attention.